Namaste students. I am JVN Dr. Aziz Muhammad Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss and continue the lecture on the genetics of immunoglobulins. In the last video, I discussed about the genes of the immunoglobulins, genes of the light chain and the heavy chains, which are found in each immunoglobulins. In this video, I am going to discuss about the how these genes are combined in each other for the preparation of a complete uh, gene, complete antibody. So, in the last video, I discussed light chain and the heavy chain genes separately. So first, we will discuss about the genetics of light chain and their recombination. So in the light chain, how many type of genes are there? In the light chain, there is a kappa chain and the lambda chain. In the kappa chains, there are variable region and the variable region genes in the J genes. There are 40 V genes and 5 uh, J genes, which are bound together with the constant region. So variable region and the constant region will attach to each other for the preparation of the complete gene of the immunoglobulin in the light chain. Similarly, in the lambda chain, there is a variable region and the J region and the constant region genes. V genes, J genes, and the C genes. So now we need to discuss about their gene organization. So that we discuss here. For the preparation of the complete genes, the VJ genes must be combined to each other for the preparation of the VJ recombinations. Here, VJ recombinations, because here only V region and the J regions of the variable region are involved. So V and J regions must be there to make a kappa chain, and the B lymphocyte lineage selects one of the VK genes, and this V genes will bind to the J segments. How this selection of V and J chain is made should be known. So what is the process of V region and the J region? What is the need of this VJ recombination? The need is that for millions of the genes, millions of the antigens, we need millions of the uh, antibodies. And for those millions of antibodies, these antibodies will directly link to the antigens. So there is a probability. They are based on the probability. If there are V40 genes, V40, the functioning of this recombination is to make the probability, make the diversity in the genes. How the diversity is made? Suppose there are 40 genes, one V40 genes, V40, V1, V2, VK1, VK2, VK3, VK4, V4, VK5, VK6, VK7, and there are G 5J genes, JK1, JK2, JK3, JK4, JK5, and then constant region genes. So the way region genes, if we talk about the diversity, how the diversity and variability will be made. V1 will bind to J1 or J2 or J3, J4 or J5. So it will be 5 diverse group. Okay, 5. Then V2 can also bind to J1, J2, J3, J4, J5, J6. So there are 10 diverse group. Now V3 can also bind uh, V3, J1, J2, J3, J4, J5, 15. V4 can bind to any of the J group, V4, J1, V4, J2, V4, J3, V4, J4, V4, J5. So they will make 20 combinations, 20 types of antibodies. So this, by this method, they can be able to make different diversity of a particular single antibody genes. And due to this sequence, they can make at various antibodies also. So we will discuss here the recombination method. Here in the Vijay recombination here, this is the double stranded DNA. This is the light chain of kappa chain. Here is the V1. Here is the V2 light chain. Here is the V1, V3, V4, V5, V6, V7, and here is the J1, J2, J3, J4, J5. Okay, so there must be a VJG joining. This is the germline DNA, which is not arranged. It is not arranged. This is the germline DNA. So it need to be joined. First, in this double-stranded DNA, selection will be made in the V region. Okay, here which gene sequence is made here. Suppose here V1 is selected. First step is selection. Suppose that here V2 is selected. For example, here V2 is selected. Okay, so V2 is must be there, and then from the J, which gene is selected? Suppose the J4 is selected. This J4 is selected. So what will happen actually? There will be a cut from here, V1 to here. So first a cut will be made here, then a cut from here, from here will be made here. Two cuts will be made here. First, the cut from V1 to V2, okay, which will remove this V1 region, and they will join the L region, 5 days region to the V2 region. Okay, from here another cut will be made to the V40 genes. So from V3 to V40 will be removed also, and it will be joined by DNA ligase enzyme. Okay, so V2 is selected here. Now time to select J4. For the selection of the J4, a cut will be made by nuclease enzyme from here to here, J3. So J1, J2, J3 will be removed out here, and then a cut here. Then a cut will be made from the J5 to from J4 to J5, and they will go to the transcription. After that, a splicing will be made here for the removal of the J4 to J5, which will cleave the J5 DNA, and it will join the C region also. So here V region, J region, and C region will combine to each other, and this is the matured messenger RNA gene. After that, this here one thing must be understand here. This is the DNA. It goes rearrangement, it goes transcription, it goes splicing, it goes translation, and then it make a complete kappa chain polypeptide. So a protein is formed at the last. What will happen? First, this is the gene arrangement of the DNA. This gene arrangement of the DNA goes rearrangement and transcription. Here V2 is selected, but J4 is selected, but J5 is not removed here. The J5 will be removed after transcription, after post-transcription modification, that is splicing. This RNA, this mature RNA will be now go for the translation process, which will make a complete gene protein here. So this is the complete kappa chain answer. This is the complete kappa chain protein molecule. So by understanding this, we can be able to understand what is the genetic organization of the kappa chain. What is the genetic organization of the heavy chain? We can also understand here. In the VTG recombination of the heavy chain, what is there? If we talk about the VTG recombination of the heavy chain, there are V region, there is a H region, and there is a J region. So we will understand the VTJ joining in the next video. So students will be able to understand the recombination of the kappa chain and the light chain here, and I hope they will better understand what is the gene organization can be made and how the mRNA and a protein will be made from a natural DNA sources after recombinations. And we can also conclude that a complete V light chain has been completed with a complete protein region. So what kind of questions can be asked here? The questions which can be asked here: explain genetics of the light chain and explain the recombination of the light chain here. So my lecture is over here. Thank you very much.